Welcome back to G Fun Facts Online. Today, we are going deep into one of the most significant paradigm shifts in modern medicine. We are looking at the operating system of cancer. For decades, the central dogma of oncology has been built on a foundation of genetic fatalism. The story went like this. Cancer is a disease of permanent genomic scars. You have mutations, deletions, and amplifications in the DNA sequence that drive cells into an irreversible state of malignancy. Under this old model, if a tumor suppressor gene like P53 or BRCA1 was mutated, it was considered broken forever. It was like a hard drive that had been physically smashed with a hammer. The only therapeutic strategy available was destructive. You had to kill the mutated cells with chemotherapy, radiation, or surgery before they killed the host. But right now, a seismic shift is reshaping our understanding of biology. We are discovering that for many cancers, the primary driver is not a broken hard drive, but corrupt software. This phenomenon is known as epigenetic switching. This reveals that cancer cells are not always permanently broken. They are often merely stuck in a pathological program. Unlike genetic mutations, which are static and irreversible, epigenetic switches are dynamic, plastic, and most crucially, reversible. This implies that rather than destroying the cell, we might be able to reprogram it. We can force a malignant cell to remember that it is supposed to be a normal, healthy cell, effectively turning off the cancer program without a single cut to the DNA strand. In this episode, we are going to explore the deep biology of epigenetic switching, the environmental factors that trigger it, and the revolutionary therapies, from EPI drugs to CRISPR-based editing, that are poised to transform cancer from a terminal diagnosis into a manageable condition. To understand this, we first have to look at the mechanics of the switch. If the genome is the hardware, the physical DNA sequence, then the epigenome is the software that tells the hardware what to do. Every cell in your body contains the exact same DNA, yet a neuron looks and acts nothing like a white blood cell. This differentiation is achieved through epigenetic marks. These are chemical tags that attach to DNA, or the histone proteins around which DNA is wound. These marks determine which genes are accessible, or turned on, and which are packed away in dense heterochromatin or turned off. In cancer, this precise regulatory system is hijacked. Epigenetic switching refers to the aberrant transition of chromatin states that locks tumor suppressor genes in an off position or locks oncogenes in an on position. One of the most critical mechanisms here is the Polycomb to methylation switch. In healthy stem cells, developmental genes are often kept in a temporary off state by the Polycomb repressive complex. This is done through a specific histone mark known as H3K27Me3. This is a plastic silence. The gene is off, but the chromatin is poised to open up quickly if the cell needs to differentiate. Cancer cells exploit this. They take genes that are temporarily silenced by Polycomb, specifically tumor suppressor genes that would normally stop uncontrolled growth, and they recruit DNA methyltransferases to the same sites. These enzymes add a methyl group directly to the cytosine bases of the DNA, creating a rigid, highly stable form of silencing. This converts a reversible, plastic state into a deep, coma-like silence. Genes like P16, which is a cell cycle break, or MLH1, a DNA repair gene, are frequent victims of this switch. They are not mutated. They are simply encrypted so heavily that the cell cannot read them. Another mechanism involved is bivalent chromatin. Stem cells possess a unique feature where a gene's promoter is marked with both an activating mark and a repressing mark simultaneously. It is like a car with the engine running, but the parking brake is on. This allows the stem cell to go in either direction, activation or repression, very quickly. Cancer stem cells hijack this bivalent state to maintain epigenetic plasticity. They keep critical differentiation genes in this state, preventing the cell from maturing into a harmless adult tissue type. This allows the cancer cell to retain stemness, the ability to self-renew and resist chemotherapy. When threatened by a drug, the cancer cell can resolve this bivalent switch toward a drug-resistant phenotype. Furthermore, the genome is not just linear, it is three-dimensional. The genome is organized into topologically associated domains, or TADs. These are neighborhoods of DNA, where genes and their regulatory elements interact. These neighborhoods are segregated into two major compartments. Compartment A is active, open chromatin. Compartment B is inactive, closed chromatin. Recent research has shown that cancer cells undergo massive compartment switching. Oncogenes that should be buried in compartment B are flipped into compartment A, gaining access to powerful enhancers that supercharge their expression. Conversely, tumor suppressors are banished to compartment B. This 3D switching is often driven by the loss of insulator proteins like CTCF, which normally delineate the boundaries between neighborhoods. When these boundaries dissolve, enhancer hijacking occurs, where an oncogene is suddenly exposed to an enhancer from a completely different gene, leading to explosive growth. But these switches do not flip in a vacuum. This brings us to the triggers. Why does the switch flip? It turns out epigenetic enzymes are metabolic sensors. They require specific cofactors derived from cellular metabolism to function. For example, SAM, or S-adenosylmethionine, is the universal methyl donor for DNA and histone methylation. Its levels are directly tied to diet, specifically methionine, folate, and B12. Acetyl-CoA is required to turn genes on, and it is derived from glucose and fatty acid metabolism. Then there's alpha-ketoglutarate, an essential cofactor for TET enzymes, which are the enzymes responsible for removing DNA methylation. Cancer cells exhibit the Warburg effect, shifting their metabolism from oxidative phosphorylation to aerobic glycolysis. This shift alters the ratio of these metabolites. For example, mutations in metabolic enzymes like IDH1 or IDH2 produce a toxic oncometabolite called 2-hydroxyglutarate, or 2-HG. This molecule is structurally similar to alpha-ketoglutarate and acts as a competitive inhibitor, blocking the TT enzymes. The result is that the cell loses its ability to remove DNA methylation. Hypermethylation spreads across the genome, systematically switching off tumor suppressor genes. This is a direct link between a metabolic error and an epigenetic catastrophe. Additionally, the tumor microenvironment plays a huge role. It is often hypoxic, meaning low oxygen, and inflamed. Oxygen is a cofactor for TET enzymes and histone demethylases. In the oxygen-starved core of a tumor, these eraser enzymes fail to function. This leads to a default accumulation of hypermethylation, a hypoxia-induced epigenetic switch that promotes aggressive behavior. Chronic inflammation also triggers the release of cytokines like IL-6, which can upregulate DNA methyltransferases. This creates a field defect, large areas of tissue where tumor suppressor genes are slowly silenced by methylation long before a tumor is even visible. This explains why conditions like colitis or hepatitis are breeding grounds for cancer. They are factories for epigenetic switching. So what does this look like in the patient? The danger of epigenetic switching lies in the plasticity it confers. A genetically mutated cell is a pony with one trick. An epigenetically fluid cell is a shapeshifter. This drives the most lethal aspect of cancer, metastasis. To spread, a cancer cell must detach from the primary tumor, crawl through tissue, and enter the bloodstream. This requires a profound identity change, called the epithelial to mesenchymal transition, or EMT. The cell switches off adhesion genes like E-cadherin and switches on migration genes like vimentin. This is not a genetic mutation, it is a reversible, epigenetic switch. Once the cell reaches a distant organ, it undergoes the reverse switch to settle down and form a new colony. Because this process relies on plasticity, drugs that lock the epigenome in a fixed state could theoretically trap cancer cells, preventing them from spreading. This plasticity also drives drug resistance. Chemotherapy kills dividing cells. However, a subpopulation of cancer cells often survives by entering a dormant, slow-cycling state known as the persister state. These cells are not genetically resistant. They are epigenetically switched into a hibernation mode, often maintained by marks like H3K9Me3. When chemotherapy is stopped, these cells can switch back to a proliferative state, causing relapse. This Lazarus effect is entirely epigenetic. Targeting the enzymes that maintain this dormancy switch could eradicate the reservoir of residual disease that causes recurrence. Now let's talk about the solution. If the problem is a reversible switch, the solution is to flip it back. This is the promise of epigenetic therapy. We have had first-generation drugs for years, like azacitidine and decitabine. These are DNMT inhibitors. They work by blocking the enzymes that silence genes, depleting methylation marks during cell division. This allows silenced tumor suppressor genes to wake up. They have been game changers in blood cancers, like myelodysplastic syndromes and acute myeloid leukemia. However, in solid tumors, they have been less effective on their own because their action is global, affecting the whole genome, and solid tumors have complex microenvironments. But we are seeing a breakthrough with precision switching. In February 2026, researchers at Monash and Harvard universities identified a mechanism to permanently switch off cancer genes in leukemia by targeting two proteins, menin and DOT1L. Menin is a scaffold protein, and DOT1L is a histone methyltransferase. In certain leukemias, these proteins are hijacked to keep pro-growth genes locked in the ON position. The researchers found that inhibiting menin, or DOT1L, didn't just pause the cancer. It erased the epigenetic memory that kept the cancer program running. The cells didn't die from toxicity. They differentiated into harmless blood cells. This represents a new class of epigenetic erasers. There is also the issue of epigenetic immortality. Most cancers achieve immortality by activating telomerase, or TERT. In many cancers, the TERT gene is switched on by a specific pattern of monoallelic methylation, where only one parent's copy is methylated and active. Even more interesting, some aggressive cancers use a pathway called ALT, or alternative lengthening of telomeres, which is purely epigenetic and relies on a specialized chromatin state. Therapies targeting the ATR kinase, or specific G quadruplex stabilizers, are currently in trials to break this cycle, but the ultimate frontier is CRISPR Epigenome Editing. Unlike standard CRISPR, which cuts DNA, this technology uses a dead version of Cas9, known as DCase9, that cannot cut. Instead, it is fused to an epigenetic enzyme like a DNMT or a TET. We can program the DCase9 to find a specific tumor suppressor gene, like BRCA1, that has been silenced by the cancer. The fused enzyme then removes the methylation only at that specific spot, turning the gene back on. This causes no DNA damage, no risk of new mutations, and is highly specific. Researchers are even using this to silence KRAS, one of the most undruggable oncogenes in history. By depositing repressive epigenetic marks directly onto the KRAS promoter using a CRISPR-based repressor, they can switch off the production of the mutant protein at the source. This is not gene editing, it is gene silencing via engineered epigenetic switching. In conclusion, the discovery of epigenetic switching as a primary driver of cancer is fundamentally a hopeful one. It implies that the malignant state is not an immutable destiny written in stone or DNA, but a reversible condition written in a code we are learning to decipher. We are moving toward a future where cancer treatment may not look like warfare, bombarding the body with toxic agents, but like reprogramming. A patient might receive a code-breaking therapy, a combination of a metabolic modulator to starve the epigenetic enzymes, a specific inhibitor to erase the cancer's memory, and perhaps a CRISPR-based editor to reawaken their sleeping tumor suppressors. In this new era, we do not just aim to kill the cancer cell. We aim to cure it, to flip the switch, restore the software, and remind the cell of what it was always meant to be. The era of epigenetic medicine has arrived, and with it, a new mechanism for cancer suppression that may finally outsmart the disease's deadly adaptability. Thanks for listening to G Fun Facts Online.